So now we are going to move on to the Q&A session for this section. And our first question is for Bill, and it's wondering whether your team has recommendations for better design to avoid bypass of the silva cells.
Yeah, that is a great question, Mike, and I appreciate Peter asking it. And for any of y'all that may have seen the typed answer that I gave Rick Savage, it does relate to that. The biggest issue is blockage. Now, how does the blockage happen? It can happen either because it's a curb cut that gets stuff that collects in it, or in the case of the Fayetteville site, it was permeable pavement that got clogged and then wasn't maintained. All right. The safest bet to place a silva cell is in a sag point. And for those of you who are like, a sag point, think about how if the street kind of goes down and then back up, that point of inflection is called a sag point. And if you were to put your silva cell in the sag point, the water should be able to get in. That doesn't mean that maintenance, you can neglect maintenance. The street line still has to be maintained. You still have to sweep. You still have to make sure you don't have debris accumulating. But if you can put it in a sag point, that's great. I talked to a colleague at UNC facilities. Some of y'all might know Sally Hoyt, and she was talking about putting silva cells in and having roof leaders come down and discharge in at the top of a silva cell. And I think that is a great idea as well, where you literally just carry that water right to the top of the silva cell. A third option for along streets is to couple the silva cell with a drop inlet, a second inlet where the water is inclined to go into that drop inlet, and then have that drop inlet serve as a flow diverter, so that low flows go into the silva cell, and then high flows would just bypass. And we spoke, in fact, with the silva cell folks, Deep Root, yesterday about the study, and that was one of the things that they thought that they were going to start promoting, was this idea of having a second or a principal inlet that the water would flow into and then be diverted to the silva cell from there. So it's a good question. It's important, obviously, if we're going to use it, that we got to make sure the vast majority of water gets in.
Thank you, Bill. That's great. And just to emphasize how fortunate we are to have your program in the state, because we're not just putting stuff out there and hoping it works. We're getting the knowledge from your group to understand whether and where it's likely to work. And that is definitely an incredible value for us.
Mike, thank you. I appreciate you saying that.
Absolutely. Okay, so Bill did a great job of talking about things he found that were a surprise. Maybe if we could get Brent and Mike, first Brent, to talk about things you found that were a big surprise to you. I love the fact that you're equating reservoirs to blue carbon. That's certainly a glamorous group they joined.
Yeah, the rates were really higher than we anticipated. We haven't worked up all the cores. This is a site near the dam that I expected to be highest in both accumulation rate and have a significant amount of carbon. But these rates, I was thinking something more like 200 grams carbon per meter squared rather than 4 or 5. So I think this has been understudied. To my knowledge, no one has really looked at these profiles with depth. They've just take a surface carbon concentration and somehow measure accumulation rates like the bathymetry maps over time to get a distant, a set of accumulation rates, which is, pretty crude compared to our approach. And so I think there's room. We're going to learn a lot more about this, and the rates are going to continue to surprise us, I think.
Following up with Brent, there's a question from Rich Gannon asking about why the annual accumulation rates would increase so dramatically through the lake's history, and whether there might be an understanding of the fade-in pattern of the nutrients associated with that organic mass.
Yeah, that's a really good question. We are uncertain if there is any pattern so far in terms of accumulation rates. It's been observed in a lot of natural lakes and other reservoirs, though there's not a lot of data from reservoirs, that there is a sort of a natural increase as a cumulative effect of watershed inputs. And what we didn't know, that part wasn't surprising. What we did not know was if the carbon would follow that. In other words, what is the source of the carbon? Is it all from the watershed? Is there a tremendous amount of productivity within the lake responsible for some of the carbon that's buried? And we're still really sifting through this, and we have a lot more workup to do for these, but that's a really excellent question.
Thanks, Brent. So Mike, to you, what were your surprises, good or bad?
I think some pretty good news would be if you look at some of the main places where we could find research on on-site systems in the Piedmont would be around Atlanta area, David Radcliffe's group, and they were showing just a lot higher septic system densities than we have in Falls Lake. So I think that's good news. You can kind of find up to maybe at the highest 3 or 4 systems per hectare, and there's definitely a signal in the stream then. But most of the sites that we're looking at, you end up having around 2 systems per hectare. And so what we're going to figure out more when we get a full year of data is sort of looking at some of the geological influence and seeing if there's clusters. So basically, if you have 50,000 systems, and some of them are treating 95% of the nitrogen, that's really good, but some of them might be just treating 70%. And so if there are clusters, there can be impacts to the streams. The good news is there's some attenuation along the stream as it moves to Falls Lake. And I think the modeling efforts will be really key to look at the whole watershed and look at patterns. So we're looking forward to that part. Yeah.
Great, thank you. And I think it's another example of that class of information where the more we know, the more we can do a little better job of thinking about space, whether it be a geological space or whether it be space on the street curb. What are we doing? Are we doing not sags, street sag, something like that, Bill? Ah, gotcha, yeah. So we're looking forward to having, definitely having lots of trees sucking up stormwater at Carolina. So let me know when you're coming, I'll help you put them in.
Okay.
So an additional question, I think, is just to think broadly about things that on the outset you didn't necessarily think would provide insights to the overall management of Falls Lake. So additional insights you've gained that maybe wouldn't have occurred to you as we came into this project and this additional information gathering. And Bill, if there's a good example, that's great, but otherwise we can.
Okay, I mean, I think one of the things that the study has shown me is that sometimes what we have been using is good, in that bioretention, which is probably the second most commonly employed practice, certainly in nutrient sensitive watersheds. If you can use that in urban areas, in suburban areas, that is probably your best bet for nutrient removal. If the target really is to get rid of nitrogen and phosphorus, if you can use a standard bioretention cell, then I think you're probably looking at your most reliable bet. So the funny thing is this is not something that we studied. We studied other sort of derivations from bioretention, but if you're trying to optimize for water quality, then I think a bioretention cell still remains your best bet in urban areas if conditions allow. Now, obviously, sometimes the reason we look at things like silva cells or sand filters is sometimes the conditions don't allow. But I think what I'd like to encourage everyone is to take a second look at sort of that standard bioretention design that's out there and maybe employ it with a little more reliability.
Thanks. That's a great example. Either Brent or Mike, do you have things that you didn't think of before you started that have really occurred now to be an insight in trying to manage the system well?
Well, the one thing that I did not anticipate that I think is going to be very useful is our categorization of inputs from the 4 tributaries. And we did that because we wanted some idea of what those concentrations were when we started trying to compare them to what was being, buried in the lake bed. We didn't anticipate them to have a response that was relatively predictable, and maybe we were a bit lucky, because one of the things that we did not have during this period of time was just a catastrophic event where we had a tremendous amount of runoff, and everything was a pretty good range of discharge, but manageable. And so it was interesting that the smaller tributaries had more of a linear response. So there wasn't a lot of area for that material to come from. And so those all made sense after the fact, but we didn't do that to have a predictive capability of inputs of those particulate materials. So that was a plus.
Great, thank you. Mike, what you got for us? Exciting surprises.
Yeah, a couple things. One thing is a lot of the stream water quality monitoring is done on larger streams. And so honing in on these small, like 50-hectare watersheds, we found some really high nutrient concentrations in some of those. And so those are kind of off the radar. So I do think it's helpful to get more and figure out why. The other thing is with having some of those new conductivity loggers on the streams, and you start to see sometimes, even when it's not raining out, there's some spikes. And so there's stuff getting into our streams. Exactly where it's coming from and figuring that out would be helpful. So yeah, those are just a couple.
Absolutely. Well, thank you all. That was we've set the bar high with the first session. So thank you all. That was excellent. We will now be transitioning to our interactive session. And so I'm going to yield to my colleague Grant to guide you to the section.